Hi, my name is Justin.
Hi, my name is Derek.
Hi, my name is William.
For the past week, us, along with all of our classmates, have been learning a lot about nanoscience and all of its different applications in the real world. And these applications can range from something as arbitrary as sculpting a house onto the tip of a pencil, or to something as world-defining as a cure to cancer, and everything in between that. For our presentation, we're going to be specifically talking about biotoxicity, which is when certain materials possess a characteristic that allow it to be toxic to living organisms. Specifically, we're going to be talking about silver, which is especially important because it's toxic to prokaryotic organisms. However, it's not toxic to eukaryotic organisms, which allows it to be very toxic against bacteria, and it's very effective in treating wounds. Scientists have been aware of this property of silver for a very long time and have been using it for a very long time. However, more recently, with the rise of nanoscience, scientists have been able to rethink and reorganize their use of silver particles to make them more effective. An example of this would be, again, with fighting bacteria. Silver nanoparticles are much more effective in the process of fighting bacteria. However, there's a lot of other examples outside of just fighting bacteria, like in wounds. An example of that would be, more recently, scientists have been weaving silver nanoparticles into clothing to allow them to become bacteria resistant, which would keep them fresher for longer and allow them to not smell. And now my partner Will is going to elaborate on this scientific phenomenon.
Okay. So I actually really like this picture because they're able to incorporate a lot of concepts into what little they had. So there's like three main things that I want you to notice. There's this grass health kind of monitoring. So up here it says mostly dead grass, and mostly alive. And this wall has 3 doorways, and 2 of them are the same size, and one of them is like very large, and I'll explain that later. And these 3 objects, so there's like a long line of people, a car full of people, which is, I know it's hard to see, and this bus that has like some people trickling out of it. So this long line of people represents the ions, like the silver ions that we were using in our lab. So basically, the silver ions, they enter through the protein channels, and they disrupt the mitochondrial functions. So they are like relatively like efficient, but not as efficient as this. So this is a car full of people, and so this car full of people represents the nanoparticles, because the nanoparticle. Okay, so I'm gonna take a breather. Yeah. So I think it's important to notice that the car is full of people, which is pretty is like analogous to the fact that nanoparticles are like a group of ions, right? So
All right. So, okay. Yeah. All right. So basically, carry on from Will. Basically, what you see here, these cars would represent the nanoparticles. Nanoparticles, as we know, are bigger in size than as compared to these regular individual silver ions. So that's why they are drawn in these cars in a bunch. And also, as we know, that silver nanoparticles are technically just a bunch of silver ions. So thus, it's drawn with a bigger surface area. Silver powder, though it may seem small to our eyes, we know that on a nanoscale, it is actually very, very large, because though we can still see it, since we can still see it, that means that it is technically not on the nanoscale. So thus, it is depicted as a bus, just like how silver powder will kind of lounge outside of a cell and then slowly dissolve, and then thus the silver ions dissolved off the surface of the silver powder will then diffuse into the cell through protein channels, and thus the slow trickling of people into the cell. So basically, these nanoparticles are naturally attracted to the mitochondria within this eukaryotic cell, because typically mitochondria are located near the perinuclear area of the cytoplasm, basically meaning that they are near the center of the cytoplasm, center of the cell, and next to the nucleus. Basically, once inside the cell, silver ions catalyze the formation of reactive oxygen species, ROS for short. ROS basically is what provokes lipid peroxidation, which is basically what prevents DNA from further developing, and thus when we did our yeast experiment, we saw that it then restricted cellular respiration, and thus no more CO2 was produced from this experiment. Along with the cellular membrane, the mitochondrial membranes are also disrupted, which then completely obliterates the cell membrane, and thus mitochondria breaks down, and thus the cell is rendered useless. And thank you.
Oh, so many for this one. All right. I will.
Hi. So, can you guys clarify the following terms? Mitochondria, lipid peroxidation, and yeah, those two.
All right, so as we all know, the mitochondria is the powerhouse of the cell. All right, sorry, I just got to put that out there. Mitochondria is basically what we know is what I think is the major center for producing ATP during cellular respiration. Lipid peroxidation, is basically a chain reaction that results in polar chemical groups being attached to the nonpolar tails of these lipids in a membrane, and thus making these hydrophobic and useless, and thus this hydrophobic balance is then disrupted, and then the whole cell membrane is just destroyed.
You said that the silver nanoparticles were only destructive for prokaryotes and not eukaryotes. What are those and why is that?
Well, prokaryotic cells are basically unicellular organisms, so they only have one cell, whereas eukaryotic cells would be multicellular, and you know the reason why.
I'm actually unsure if they are harmless to eukaryotic cells. I'm not sure about that one.
Okay, so one of them has one cell, one of them has many cells, right? So if you kill the one cell, how does that disrupt the many cells?
So you mean, like, for example, destroying the mitochondria in the eukaryote, or?
Yeah.
All right, so if you destroy the mitochondria in the eukaryote, then it would render the entire eukaryote as useless as well then, right?
It would lack the means to produce efficient energy for the cell because it's losing cells that could produce the energy that it needs, I would think.
Okay. So it's just like killing one, but like when it's a eukaryote, there's a lot of cells, right? So it can't kill all of those cells. Does that make sense?
Yeah.
Yeah, okay. Thank you.
We should have asked
Talk to us about this.
Hi.
Hello.
I was going to talk about the silver powder.
Mm-hmm.
Could you please describe a couple of factors that affect the rate of diffusion of those ions?
Okay, well, thank you for giving me this opportunity to redeem myself. So basically, the silver dust, they're like these clumps of like silver atoms, right? And you can see it by the trickling is analogous to like that slow like dissociation of the silver ions from the surface of the dust particles. So that's why they're the most inefficient at like disrupting the mitochondrial functions.
So if you're comparing the surface area, because surface area is always the thing that we love talking about with nano, how would the surface area compare between the nanoparticles and the powder?
I'll take this one. All right, so basically, nanoparticles, the surface area is much larger, and thus that's why we see here on the little grass diagram, it says dead grass, because it basically symbolizes that it does the most damage. Rather compared to silver powder, it's just a round surface, thus it doesn't cover that much surface area. So it's relatively just mostly a live grass.
So for the same volume of powder versus nanoparticles, if there's more if there's more atoms on the surface of the nanoparticles, more of them can dissociate more quickly, right?
Right.
All right. Good job. Thank you.
Thank you.